Okay, this is the center. Actually, we are very close to the Basque Center for Language Studies. That is the previous presentation by Cesar. In the same campus, in the hill, on the top of the hill of San Sebastian, a little bit far from the beach. But yeah, this is the building. I'm directing the molecular and functional biomarker groups. I moved to San Sebastian about 4 years ago. I also a member of CIBERES, which is a Spanish network of researchers in respiratory diseases. This is the name of the center, SIC Biomagune. It's a center devoted to the study of biomaterial in general, with different applications, also imaging, as you will see in our group, another group like Pedro Ramos, who actually is one of the other group leader in the center, but mainly working in the brain. And the other imaging people are more related to positron emission tomography application. Okay, this is part of the facility. We have a cyclotron in house that allow us to do all kind of radio tracers in the center. There is a radiochemistry lab. And they're all. This is a little bit old already because we already have a new equipment for positron emission and single photon emission tomography. We have 2 PET scanner and 1 SPECT scanner for a small animal, and we have a 7 Tesla and 11 Tesla MRI system, Bruker system to do MRI. We also have an animal house facility accredited by the ALAC. And that means that we have all the certification approved for working with different animal models. We can receive animal from other parts, and we can do experiments in different organs as we have experience. For instance, in my case, in the heart, in the lung, as you will see, Pedro in the brain. We are basically covering everything. Danny Padró, who is the responsible of the platform, of the MRI platform, is also a very well expert in different applications. Okay, this is also because it's a nanoparticle devoted facility. We have everything in-house to characterize this nanoparticle before injection. We have also a very nice laboratory for microscopy, optical microscopy, and we are thinking the possibility of including also fluorescent tomography in the center these days. As I said, there are different labs to prepare radiosynthesis. It's a unique center in the center. It's part of what is called the ICTS, the Singular Center for Research in Spain. It's distributed with CNIC in Madrid, Universidad Complutense de Madrid, and the Hospital La Fe in Valencia. Okay, my group is basically working in imaging-based biomarker, but we are doing a lot these days in 4D flow, for instance, a hemodynamic-based biomarker. 4D flow not mainly in small animal, but in human studies. And we are also doing a lot of metabolomics, NMR-based metabolomics, and of course, multi-scale imaging. In 2 diseases, vascular diseases, that's why flow is have an important role. We are working in pulmonary hypertension. We have different models. We are also working in atherosclerosis. Pulmonary fibrosis is also part of the research that I'm doing in collaboration with Susana Carregal, who is one of the recent Ramón y Cajal fellows who joined to my group. And there's some cancer, but not in MRI, in relation to MRI, but into PET. Of course, is the important thing in these 2 diseases is the study of metabolism and the relation of metabolism and disease. And this is part of the experiment that we are doing. This is, for instance, an example of the things we are doing in nanoparticle. This has not been published so far. This is a model of atherosclerosis. We are visualizing by MRI the aorta, and this is the previous and post-injection using calcium carbonate modified nanoparticle with alendronate on the surface just to target calcification, microcalcification. This is ongoing. We have additional result that if you are interested, just let me know and we can discuss about that. These are recent publication, very recent publication. There is a publication of my group in pulmonary hypertension in collaboration with the group of Jose Antonio Enriquez's CNIC, the Spanish Cardiovascular Research Center, and we characterize a group of mice that have metabolic disorders and also mitochondria-related diseases. At the end, we related to a pulmonary hypertension after characterization by MRI, for instance. This is another application of the nanoparticles field, not only for MRI, also by PET, because we are modifying the nanoparticle, the iron oxide nanoparticle, with a PET tracer just to quantify and study the biodistribution of this tracer. This is a very nice radio tracer with many possibilities by MRI because they have a relaxometric property interested, and also the catalytic has also have catalytic property. We demonstrate in this publication. You can find the detail, but their application with as also as anti-cancer therapy. And this is one of the study characteristic of my group. I studied basically the lung many times, and pulmonary hypertension is, for instance, one of the diseases, as I said before, fibrosis, as indicated in the collaboration with Susana. We are investigating the protein corona after interaction with, in this case, with the protein surfactant A protein in the lung, because this is iron oxide base. We need to increase the signal on the parenchyma, and we start using UTE, ultra short echo time, to just visualize even iron oxide that signal on top of the background in the lung. This is the group is a little bit different these days, but this is the most recent picture I have. It's becoming a large group and they have a lot of support from different institutions. Thank you for the attention. I'm open for discussion. Thank you very much, Cesar.